give quite a lot of talks to embryologists, and the first question that always comes up is, am I going to lose my job? What's my job going to look like in 10 years' time? So I really think there'll be more jobs created in the future. They may be, as I said, you may be playing a different role. The role will change. So I think, it's hard to predict, but the position will still be there, but it will evolve in some way.
What will the IVF lab look like in 5 years? Trying to shape the response to that question as much as answer it is Boston IVF's Chief Scientific Officer, Dr. Denny Sakkas. In addition to running Boston IVF's labs and having ran labs in England and Switzerland, Dr. Sakkas is now the head of the Scientific Advisory Board for a venture called AutoIVF. I asked Dr. Sakkas about the potential downsides to automation in the lab, how his new venture works, and what are the differences between it and Aura from Conceivable Life Sciences. Denny's prediction that all IVF labs will have some version of time-lapse imaging in 5 years, when and where embryologists need to stand firm about what type of conditions or supplies they have to work with, other innovations he's excited about, and what he says he plans to buy for Boston IVF's labs within the next year. Enjoy this episode about automation and chain of custody management in the IVF lab with Dr. Denny Sakkas. If you like the information in this episode, we're compiling so much more data on new technology in IVF, products, services, market analyses, info on mergers and acquisitions, and data on every company in the fertility space. Go to data.insidereproductivehealth.com. If your company isn't an IVF Heroes Universe sponsor, your competitors are so much more visible to your customers because we report on your category and we give that info to your customers. Go to data.insidereproductivehealth.com to learn more about other fertility companies or submit your company today. Don't be left out.
Today's advertiser helped make the production and delivery of this episode possible for free to you. But the themes expressed by the guests do not necessarily reflect the views of Inside Reproductive Health nor of the advertiser. The advertiser does not have editorial control over the content of this episode, and the guest's appearance is not an endorsement of the advertiser.
If you think about it, we've had automation for a long time. Some of the older embryologists, and maybe I'll include myself in that ilk, we used to make their own culture media, for example. We used to make our own micropipettes for micromanipulation. Automation basically took that away from us in some way because automated companies, well, if you want to call it that, but companies now are automating that process. So we buy all our culture media. I'm not really aware of any clinics now that make their own culture media. Sadly, I think if you ask most of our embryologists, they wouldn't know how to make even culture media. So that's been automated for a long way, even though it's automated by commercial providers and we buy it. And micromanipulation pipettes, they were a pain to make. I can guarantee you that. And now, the companies have automated that. There's quite a few companies that provide them. They know, when we buy a pipette, we know it's going to be the right diameter, the right angle, the right consistency. And I can guarantee you many, many years ago, it wasn't like that. So in some forms, automation sort of has been around for quite a while. I think now we're starting to look at it slightly different in terms of how it's sort of coming into the process.
You said, sadly, most people wouldn't know how to make culture media today in the lab. Are you just waxing nostalgic when you say sadly as a figure of speech, or do you feel like we did lose something by people not having that practice?
I think it's maybe a topic of another podcast you might want to do, but the change in our field, many years ago, the embryologists were all PhDs. They'd come out of animal backgrounds, and I'm talking 30, 40 years ago. And just the growth in the area has demanded that we have, you don't have enough people with that training. So a lot of the embryologists now that are getting trained, I don't want to call them technicians because they're really clinical embryologists, but a lot of the background that, people that have been in the field for 30, 40 years is missing. And little things like, well, not little things, but understanding culture, how you make culture media, how we used to make pipettes, that's changed a lot with the implementation of, being able to get these products. And that's happened in all fields. Genetics is the classic. I don't think half of your molecular biologists running genetic assays in the lab probably know how to make some of the buffers and some of the materials that go into running the genetic tests. So I think it's happened across the field everywhere.
Now, my understanding would be that if people are doing less of that, if they're not having to put pipettes together because they're a pain to make, if they're not having to figure out how to make culture media, that they are becoming less of technicians, and then they're able to free up their time for more study, for more experimentation, for more research, to become more of scientists and less of technicians. But is that not necessarily the case?
I think it could happen depending on, the personalities in the field. So I think that definitely, has occurred in the past, and hopefully it will occur in the future. And we're talking about automation. One of the good things, hopefully, of automation, it will allow the embryologist to do other things, to be more focused on certain procedures that are, much more difficult. Maybe be to sort of have more patient interaction, which will be, a better thing. And obviously, hopefully, think about things that will improve IVF in the laboratory, especially, which is sort of the area I'm involved with. So I think you're correct that, hopefully it will allow certain people that have that drive within them to have more time to do things like think about how do I improve the process, and what else can I do to make things better for the patients.
Do you think that patient interaction with embryologists is an inevitability? Is it something that we're just starting to see a little bit of now? Yeah, we had Professor Christina Hickman or Dr. Christina Hickman on the program, who runs a program called Avenues in London. And then I've seen some other folks talking about using embryoscope for the reason of being able to show patients, here's what's going on with your embryos. But it's the embryologist that is having some contact with the patients, at least in some programs, at least in Dr. Hickman's program. Is that something that you think will become the standard, or are you not so sure?
Historically, the embryologists had a lot of contact with patients. They would do a fertilization call. They were more in touch with them about, how their embryos are growing and things like that. So if patients had a question, they would sometimes, contact embryologists to talk about it. That sort of it's stayed in a few of the smaller clinics, but I think the larger clinics, it's sort of, they're just too busy and it's gone away. So I would hope that having a bit more, not downtime, but having some automation would allow that. And even as you mentioned, automation like being for patients being able to access their embryo videos or embryo pictures through patient portals, and maybe more interaction through patient portals, which are secure, allow that. So I would hope in the future that embryologists do have a particular place where they are able to interact more with patients, because in the end, I think a lot of us get into this field because of that want and feeling that we're really helping patients. So if you're not talking to them, you lose a little bit lose a bit of that. So I know, and I still do. I still enjoy talking to patients when I can.
So I want to talk about those benefits that could come from automation, because maybe those are among the duties that embryologists are able to pursue, and perhaps more research and other things they can do when they don't have to be doing so much manual work. I do want to ask, if in your view, is there a risk to automation? Can you foresee some downsides or some unintended consequences, some second or third order consequences that, if we're not careful about, even if they don't outweigh the benefits that automation would bring, that you still wouldn't want them? Are there some things that you're concerned about?
Well, I mentioned before that it may help us sort of interact more with patients, but it may remove that personalization from talking to patients, and it there is a risk, as much as we don't really want it, that um, the the true clinical embryologists that we have may become a bit more technical in some way, and not have the background about talking to patients, understanding basic embryology. So that's one of the risks, that it may become just a technical expertise or move more to a technical expertise. So that, I think, is always one of the risks with automation. But as you said, I think that may not happen, or it may allow embryologists to sort of pursue maybe a more technical career, but others to pursue more of a research, or a more clinical embryology focused career. So it may in some ways separate sort of the type of people that we have in a laboratory. It might be different roles that they may play in the future.
Your sample size might be skewed, the question I'm about to ask, with what you're hearing from embryologists, because you're working on automation. So maybe you're talking with folks who are more excited about it. What's your litmus test of feedback from embryologists on automation? My guess is that it would generally be good if they can see these other opportunities, because I've talked to so many young embryologists. I'm talking folks in their mid 20s, late 20s, that want to leave embryology because they just don't want to be in a lab all day. They don't want to be in a 10 by 12 room or whatever it is, and feeling like they're just going back and forth from station to station, and no windows and no ability to work from home, et cetera, et cetera. And so to me, it seems like, okay, if they could be doing other things while there's robotics and technology in the lab, that they'd be favorable to it, but maybe not. Maybe I do see some people on LinkedIn especially that like to comment that they're very skeptical of it. What's your straw poll of what embryologists are saying?
I obviously give quite a lot of talks to embryologists, and the first question that always comes up is, am I going to lose my job? What's my job going to look like, in 10 years' time? Firstly, automation and any new practice or new equipment or whatever always takes a long time to develop. So, there's a timeline of when these things will come in, that might be longer than, all of us think. Secondly, I tell them that automation, one of the things we hope it does is bring a greater access for patients. So, in many ways, the labs might be much busier than they are today. So, that may not be a good thing if someone doesn't want to work that much, but I'm sure they will be busier. So I really think there'll be more jobs created in the future. They may be, as I said, you may be playing a different role. In terms, you may be running different types of equipment, may have more of a interface with computers and interfaces with instruments, if you want. The role will change. I don't know if it's going to be, you're going to be locked up in a room in a 10 by 12, hopefully not. And hopefully the automation will allow you to get away, you know, for 10, 15 minutes, rather than fully having to concentrate on a particular technique. And, you will have 10, 15 minutes to get away, maybe do administrative work, do other work. So as I said, I think the role will change. I mentioned before that, a lot of embryologists used to spend a lot of their time making micropipettes and making culture media. That we forget about that, but we're still busy and we're still able to do other things. So, like all these things, I think the role will evolve. How that will evolve? Will it evolve a more technical? Will you be more of a technician, or will you have time to do more research or think about novel ideas? Have more time to interact with patients. I think it's hard to predict, but the position will still be there, but it will evolve in some way.
Tell us about the project that you're working on with regard to automation.
Yeah, so, with good or bad, they say that, if you keep repeating the same thing, you might be either crazy or brilliant. I'm probably more on the crazy side. I've been involved with a few startups for quite a few years, going back to the early 2000s and even before that, actually. The current project I'm involved with is, we're really excited. It's a startup company called AutoIVF, and I'm lucky I'm chairman of their scientific advisory board. This is a novel technique based on microfluidics. Microfluidics, I'm calling it a novel technology, but it's a platform that's been around for many, many years actually now. I'm lucky to work with some really amazing people that are in that field, and it's been used for many years for isolating, rare cells from, blood, basically. So you can it's quite amazing. You can find one rare or two rare cells from blood in particular cancer diagnosis from, liters of blood to detect cancer. So that's been around for a while. So we've actually, in collaboration with the people we work with, we've used the similar technology where you can isolate from large volumes, and the volumes that I'm talking about are follicular fluid. So what we've developed is a system that actually can find the oocytes in the follicular fluid, and it's completely automated. So you pour the follicular fluid in one side, the device actually sorts through all the blood, the tissue, the somatic cells, finds your oocytes, and then it will denude your oocyte. So from 100, 150 ml of follicular fluid, you end up with a few microliters of very clean oocytes at the other end in a very 15 to 20 minutes. So it's quite exciting. And the company also has other techniques in the pipeline, again using microfluidics for doing other processes in the IVF lab.
And what was the genesis of deciding on this as opposed to any number of different other approaches you could have taken?
I think it's the team that we had developed. It's a very strong team. Obviously, their focus was microfluidics. We also wanted to not repeat what's going on in the lab, so not sort of just mimic all the steps that are going on in the IVF lab now. We wanted to introduce novel concepts. So, and actually, the technology has brought some amazing surprises to us in terms of just simply, trying to take a novel approach at an egg retrieval process. The egg retrieval process, I'm sure in 1978, the way Bob Edwards did it, then it hasn't really changed that much. You put the fluid in a dish, you look around, for cumulocyocyte complexes, you clean them and put them in, in a new drop. So we wanted to challenge those concepts with novel technologies and with new technologies. And I think we've done that, having some of the results that we've seen already.
So what do you think will be the wider application of this? Is this something that labs need to buy additional equipment for, or do they need to change the space of their lab in any way? Do they need to change their workflow in any way?
No, actually, it's something that will fit into their workflow. So every lab, every IVF lab does an egg retrieval. It's the device is probably the size of a printer. Not many people use printers now, I think, even so. It could replace the space that you had your printer in. So it's just a little bit larger than a just genuine printer. It could sit in your egg retrieval room. It could sit where you're actually currently doing your egg retrievals. So it basically will allow you to, pour the fluid in, whether that's an embryologist, a technician, even a nurse in the operating room. And, 20, 30 minutes later, depending on the type of retrieval, you will have a dish ready for the embryologist to take those oocytes and continue, to do ICSI, to do egg freezing, whatever. So it basically will help the workflow. It'll take away the embryologist's job of having to concentrate there and doing the egg retrieval process. And as I said, there's some added benefits that we've already seen to this process.
Are there still decisions as this standardization happens and automation happens across the lab? Are there still decisions that really should be being made at the local level? So part of the promise of standardization is you don't want so many darn decisions made at the local level because there's so much variance, and with that variance, it's hard to do quality assurance and quality control and come up with best practices and follow the scientific method. And so you want to come up with, here's the best practices, and then we replicate those best practices at scale. But are there still decisions that should be being made by the embryologists as these things become automated and standardized? What do embryologists still need to be in control of at the local level?
I think just the process, the logistics of the process, so handling the material, making sure the chain of custody is correct, making sure, that the quality control of all these instruments, we have a lot of instruments in the labs already. One of the things we're very pedantic on is that the temperature's correct, the gas environment's correct, that the eggs and the embryos and the sperm, are very precious, and that we're treating them correctly. In effect, we're chaperoning them from the ovary back to the uterus in some way. That's the job of the lab. If they're good, we're quite good now at getting pregnancies, establishing pregnancies, but definitely in those 5 to 7 days, and obviously freezing, etc., you can do a lot of things wrong that will harm the chance of a patient's, pregnancy chances. So I see the lab as a very strong chaperone for this process. And I think still, whatever we do, whatever automation we introduce, see the role of the laboratory, the embryologist, is in making sure that sort of piece of chaperoning is consistent. And like you said, that we're doing it the same in all labs around the world. Unfortunately, we know that probably isn't happening. There are some labs of better quality than others, and there are many other biological reasons too that, there's variation in labs also.
In order for tools to actually improve efficiency and deliver real value, they have to improve your profit margins and simplify your workflow. Clinics like Twig Fertility, expanding from Toronto to Vancouver, and Care Fertility, the largest network in the UK, expanding into the US, used EmbryoScope for that. Time-lapse technology is helping IVF labs across the globe reduce human error and improve embryo viability by minimizing handling and fatigue-related risks. Dr. Sheila Ali, Scientific Director of IVF, Andrology, and Embryology Laboratories, champions innovations like EmbryoScope that enhance consistency and success rates in embryology labs. She says that by standardizing protocols and integrating automation, she is not only improving efficiency, she's directly improving patient outcomes. You don't have to take their word for it, though. You can participate in their Seeing is Believing program and trial EmbryoScope for 3 months free of charge. Why not give it a shot? Contact Vitro Life and ask if your lab is eligible for a free 3-month trial of EmbryoScope. As consolidation happens in the field, I see a bit of a spectrum. On one end of the spectrum, you might have a network that they make decisions very centralized. And if this is what we're doing across the board, we're doing it at every lab, we're doing it at every clinic. And then there are others where they're still very much kind of letting this clinic do it their way and this lab do it their way. I do worry about clinicians not being able to practice the way that they want to. What things of yours do you feel very protective about, that I want to be able to order this, I want to be able to buy this, or fire this person, or hire that person, or build this way or not this way, that you really want embryologists to stay in control of?
Yeah, that's an interesting question. I think it expands, as you said, to the clinical side too. The fear, or is that you get these big conglomerates coming in, and they focus somewhat on finances in a way. So the concern is, both for clinically and in the laboratory, that they believe a cookie cutter method of treating patients will work, and you can do that for all patients. So they sometimes may remove flexibility from either the clinician or the embryology lab. The cookie cutter approach probably maybe works for 70-80% of the patients. We do pretty well with those. But then you've got 20 to 30% of patients that may be more challenging, let's say. So there's still a lot of fundamental arguments. I won't go into the clinical side about stimulations and that, but even in the lab, fundamental arguments about techniques that we do in the lab. And again, PGT, is it good for everybody? ICSI versus insemination, is that good for anyone? Things like fresh transfer versus frozen transfer. So we're still struggling with a lot of these questions, 40 years after the first baby, basically. Making us do things in a particular way may change the flexibility of, how we treat patients. And as I said, I don't think it's probably going to hurt, 70 to 80% of cycles, but having some flexibility maybe for 20, 30% of patients could mean, for that particular patient, if they have a live birth or not. A difficult patient, that may benefit from a fresh transfer, for example, may not benefit from a frozen embryo. They may not have enough embryos, but we still we're not 100% convinced that, maybe a patient's sort of poorer looking embryo, let's say, that we might put back as a fresh and we may not have frozen, we know that they sometimes can give live birth. So I think there are things that we still need to be cognizant of and have some control, and that comes down to media, sperm preps, the ability to transfer maybe a fresh versus frozen embryos, doing PGT or not. Would be nice that we still have some flexibility in treating patients, in the future, and not maybe be told that, this is what you're doing. You're just doing things in one way, and that's the way that works. Good, but I don't. I think some percentage of patients may lose out if we take that approach.
I think you've listened to this show before and you know that I'm not an embryologist. I don't have a scientific
Yes.
background. So I can't judge if how significant the quality of oil matters or the quality of media matters. Who's out there that has good quality, or does it matter? Is it relatively substitutable?
One of the good things that came out of the commercialization of IVF media, let's say now as an example, and oil, is that the processes they use are very stringent. Okay, we've had some, deviations, let's say, but in general, when we buy culture media from whatever company, that we're pretty confident that that media has been well controlled, made with good medical practice, good conditions. So I think all the companies do a pretty good job now at doing this. And as I said, oil was like was like the biggest phobia of embryologists. If you had one batch of good oil, for example, we would hoard it like to a sort of maybe a very strange state that you would, lock all your good bottles of oil in a cupboard and not let anyone touch them. So, but now we're, much more confident. So all the companies, do a very good job. Historically, some of the media, I was lucky. I worked with David Gardner many years ago, and we had sort of developed the origins. It's probably changed 100 times more now of the Vitrolife media. But so I'm a bit more familiar with those. But in general, all the culture media that are being made now are, are very high quality, well tested, and I think most people can trust them, I think.
One area where I have noticed a discrepancy between what lab directors say they want and what the business seems to be paying for is time-lapse imaging. I've asked every lab director, at least in recent memory, maybe earlier on I didn't, but I've been asking them, do you view time-lapse as a nice to have or a must have? I think all of them have said either must have or quickly becoming a must have. Maybe there's somebody that says nice to have, and maybe you'll be the contrarian that says it's just a nice to have. What's your view on time lapse?
So the best description I've heard about time lapse is from Michael Alper, our CEO at Boston IVF. He calls it pornography for embryologists, and he's right. I can still sit and look at these time lapse images, the videos. They're really they're amazing. Um, I think we've always wanted to watch the embryos in some manner. So I think having the time lapse is a huge bonus in the labs. Like all new items, it becomes a commercial thing. The cost versus the the benefit. All the studies we've seen today indicate that the benefit is actually in that these time lapse incubators are very good incubators. So they're very good at growing our embryos, at taking care of them, allowing us not to move the embryos and being able to see how embryos are progressing. So in that manner, they're fantastic. I know, people have discussed AI, artificial intelligence, machine learning. At that level, we're still sort of, trying to understand how much that's going to help us. We still do quite well with blastocyst morphology and picking the embryos. I think eventually in 5 years' time, I think all laboratories will have some concept of time-lapse videos or time-lapse incubators in their in their laboratories. It may not be what we have currently, the embryoscopes and the other types of time lapse systems. So it may not look like that, but I think we will all have time lapse imaging capabilities in our incubators and our laboratories, I think in 5 to 10 years.
Why? Why is that important?
Well, I think, as I said before, one of the difficulties is we do get that information. We'll get a nice blastocyst, and a lot of those blastocysts are great. We can biopsy them, get, euploid embryos. But again, focusing on patients that may not have performed that well, we can then go back and look at their videos and say, okay, this is what we've seen in this patient. They've had delayed fertilization. Their cleavage was not, in characterization. So getting that information, getting the time-lapse imaging information for again, the majority of patients is probably not going to change that much for that patient. But again, for, your patients that are having issues getting to a live birth, having a successful treatment, we may see things in those videos that might tell us, wait a second, there's something, that with the embryos of this patient. Now that may mean we tell that patient, you may want to look at another approach to IVF or to achieving having a baby at home, basically. And that hopefully will quicken the diagnosis for that patient. So they're not doing multiple attempts of, 3, 4, 5 IVF cycles, which are, very draining on a patient's, morale, and it's very difficult for patients to go through those treatments. So I think the time lapse will also help us in giving more feedback to patients in terms of their embryology, their embryo development.
Is it possible to fully automate the IVF lab without time-lapse imaging?
You probably could, but I think, again, I think we do get a lot of information from the time-lapse videos. So I think if you're automating, if we're going, as I mentioned before, the AutoIVF system has an egg retrieval, automated. If we can link that with an embryoscope, which, we're already thinking about, with and then, the whole process is, I think, why wouldn't you, if you're automating, why wouldn't you want those videos, especially if it's, the capabilities are already there, the incubators are really good. I think we will do that, because that will be extra information that we will get. I think in the long run, even though artificial intelligence probably hasn't, given us the specific embryo morphology picture, I think having all the data, having a lot of data, including patient data, maybe other information from culture media, I think time lapse will actually help us, going forward in the future with more information.
Do you have time-lapse incubators in your labs?
So we're a very big lab. So we actually we had one, but we're actually looking at getting some in now. As I said, I think inevitably, I think down the road we'll be getting them. Historically in the US, time lapse has sort of been less, I don't want to call pervasive, but utilized because of our, a lot of labs rely heavily on genetic testing, PG. In Europe, they're much more, in Europe, in Asia, in Australia, you probably see more time-lapse instrumentation, but I think in the US also they'll be coming in soon. And we're, similar for us also, we'll probably start using them also.
So the trend seems to be moving towards time lapse. It seems, from my lay point of view, that EmbryoScope has a slight lead in that market. That when I ask people, it seems like there's a slight preference towards EmbryoScope. I imagine you're checking out them all and you're looking into them. Have you looked into EmbryoScope, and what do you see, good, bad, or neutral?
I know the EmbryoScope much better. They were, first to market. We had historically had a lot of involvement with the initial company, Unisense, that had developed the EmbryoScope. So we knew them very well. And as I said, they've probably been on the market the longest. So like all instrumentation, it's gone through its development, and it's probably, I don't want to say the most mature, but it's, it's the most common one. So I think people sort of gravitate towards that in a way for, if you're automating the time-lapse system, if you want.
So we're talking in late 2025. Maybe this recording will air in late 25 or early 2026. But if we were recording again in late 2026, think you'll have a time-lapse incubator?
Yeah, I think so. Yeah. Yeah, we'll definitely have them. And hopefully we may be doing the retrievals automated also.
Tell me more about that.
As I said, for us, that's an incredibly exciting technology. I can't tell you everything about it, but the approach that we've taken where it is a novel technology and we're not sort of relying on mimicking systems that we already do, it's actually allowed us some surprises. So we're super excited about this technology. We mentioned some of the benefits of automation and any of the types of automation that I think are coming out now. One thing it does do is it homogenizes the treatment of eggs, embryos, freezing. It sort of does standardize that in some way, which is, a thing that we worry about a lot in the lab, making sure everyone's doing the same protocol. So we're pretty excited that at least at this first step. The other thing that I think is interesting about going to the retrieval step for automation is that one of my collaborators called it's the gatekeeper of IVF. If you want, you have to get the eggs. So one of the things, and I think I mentioned at the beginning, we spoke about like automation. There's some good things and there's some things that we're concerned about. But one of the biggest things I think automation will bring, and hopefully this device that we're talking about from AutoIVF, is that you can then take that device and do a retrieval anywhere in the US. You can go to the smallest little town in the US and do a retrieval. You can freeze those eggs, ship them to the big lab. So in doing that, you're actually taking, the lab to the patient. And I think that's the big thing, for the future, that we will then increase access of IVF for, majority of patients who are infertile that don't actually have that access today. So I think that's the biggest benefit that at least we hope, will bring with our technology.
Picture this, you're running one of the busiest fertility networks or IVF labs in the country. Demand is high, staff are stretched, and every minute counts. That was UCSF until they restructured their entire workflow with the help of EmbryoScope time-lapse imaging. The result, they didn't just shave a few minutes. They are now saving 7 embryology hours every single day. More transfers, faster ICSI, sharper embryo selection, less risk, and the staff happier, less stressed, more focused. UCSF proved what's possible. Now it's your turn. Reach out to Vitro Life and ask if your lab qualifies for a 3-month free trial of EmbryoScope. It might just change everything. So forgive me for not knowing the life stage of auto IVF. I'm only slightly familiar with this venture. Are you all in commercialization stage yet? Pre-commercial? Are you doing this in conjunction with all of the pre-commercialization steps with Boston IVF? Tell me about that.
So it's we've actually we have a full prototype. We have a few full prototypes that we're now starting to put out to clinics around the US and internationally to do the next step of validation. So the company was in stealth mode for quite a few years. It's now sort of coming out, if you want, as companies do, and now making the instrument available to a number of clinics for clinical validation. Like you said, that's part of the whole commercialization process.
Have you been down to Mexico City to see Conceivable? And if so, where do they converge or diverge from what you all are doing?
Yeah, so yes, I have seen it. I've seen the full Aura system. I haven't seen it actually operating, collecting oocytes and running cases, but I know they're doing a clinical trial. It's an amazing set of instruments. I'm an embryologist, so I'm very very easily impressed by the engineering that's gone into it, which is pretty impressive. It's, as it's a series of five large instruments that are robots in a way. So I could imagine someone in the in the early 1920s seeing a car manufactured by hand and then seeing robots coming into it, and being able to manufacture a car. And so, what's great is they've taken the lab process and taken all the manual processing and use robots to do everything, which is pretty cool. They've got some other innovations which are, like the freezing technologies are very interesting. So it's a really, impressive system, but I think where we fit in, we're obviously using a completely different technology. I don't want to say, well, in some ways it's a bit more novel. The robotics has been around for many years. So, we do fit in with their system, in terms of maybe in the future, if they're set up in a large warehouse system of a lab that's more centralized, we could definitely feed oocytes to them to process and then, bring them back to be transferred if needed. We also, in some way, we're competitors. AutoIVF is a competitor. They're also developing the lab eventually. It's already, we know that we'll be in a much, much smaller footprint than what they basically have currently developed, although that, I think in a few years will probably change in a way.
What other innovations do you want your colleagues to adopt in the coming years? Do you think about what's in the pipeline, or maybe what's currently available but many of your colleagues haven't adopted yet? If you could do a Jedi mind trick with your colleagues and get them to do what you wanted to do, what innovations are they implementing in the coming years?
I spoke before about our system, even Conceivable system, hopefully will change access for patients. So the innovations I really want to see, and we're already seeing some of these, we've seen at-home semen testing, for example. There's a lot of effort going into at-home hormone testing and even ultrasound testing. So allowing the patient to do things, more in their privacy. Reproduction and infertility historically has been a really emotional thing for patients to deal with, I think. Has some taboos. I think some of those have, been lessened in a while, but many cultures, we're lucky in the US in some way, but many cultures, there's still a taboo to infertility. The more we can take things back to the patient, and whether that's testing, allowing, collection of the samples at home or closer to where they live. I think the technologies that I really want to see in the next few years are at-home ultrasound, at-home hormone testing. I'd love to see retrievals taken to the doorstep of patients so they don't have to travel, hours sometimes for some patients, or even if you're in Boston, might take you an hour to get to your local clinic to have a blood test. In New York, it's the same thing. So if we can take the treatment more to patients, allow them to do it, in a more comfortable state, I think, the stress will actually come down, and, we may see improvements in live birth rates and pregnancy rates just from allowing, a more friendly procedure for these patients. Because I don't know, Griffin, if you've been involved with IVF at all, it's an emotional roller coaster, right from the beginning of your diagnosis to, maybe even having the live birth. It really is difficult for patients. So if we can change that in a way by making it more accessible through various technologies, that's what I'd really like to see in the future.
I'm glad you mentioned that, because, and I think of David Sable and Abigail Cyrus' 3 criteria for innovation in IVF. They're thinking reducing cost to baby, reducing time to baby, and reducing life disruption to baby. We often talk about the first one, sometimes talk about the second one. Third one probably don't talk about enough, which you just mentioned. And it just can't be understated how disruptive it is to have to leave work, to have to drive across town, to have to get a babysitter, to have to, et cetera, et cetera. And I hope that the innovations that you're talking about and others really make a dent in that in the coming years. Dr. Sakkas, I look forward to having you back on the program. Thanks for coming on and sharing your thoughts with us today.
Thanks, Griffin. It's been a pleasure. Yeah, you're right. David Sable's been talking about this for many, many years, and we're following in some way in his footsteps. But it takes a village, as they say. So hopefully, we're part of that village and can get. It really is true that the access is one of the missing pieces. So the better we can get at that, I think in the future, hopefully we'll be back in a few years telling you we've got technologies that are creating that access. So I look forward to talking to you again.
Go to data.insidereproductivehealth.com. That's where we're compiling so much data on new technology in IVF, products and services, market analyses, info and mergers and acquisitions, every data point that we can on the field writ large and the companies in the fertility field, we're compiling there. Go to data.insidereproductivehealth.com as we build that over time. And if your company isn't an IVF Heroes Universe sponsorship, Your competitors are getting more visibility because we report on your category and we give that to your customers. We do that multiple times per year, and we do it around big events like ASRM and ESHRE. Go to data.insidereproductivehealth.com and submit your company today. Don't be left out.
Today's advertiser helped make the production and delivery of this episode possible for free to you. But the themes expressed by the guests do not necessarily reflect the views of Inside Reproductive Health, nor of the advertiser. The advertiser does not have editorial control over the content of this episode, and the guest's appearance is not an endorsement of the advertiser. Thank you for listening to Inside Reproductive Health.